All right, now we're going to look at the electron transport chain, right? So as you see this big mesh of just mechanisms, what's going on. I'll explain that very simply in the way before we get started. So everything we learned so far, from glycolysis to the pyruvate dehydrogenase complex to the TCA cycle, whatever is remaining from some of these metabolic byproduct enters the electron transport chain. All right, and it takes place in the mitochondria, specifically in the intermembrane space and the matrix. And so the way it first gets controlled is through this theory called the chemiosmotic theory. So the chemiosmotic theory is looking at the different gradients created by these hydrogens. All right. So in majority of the cases, you look at the intermembrane space in the matrix, majority of the hydrogens, ions, right? So these are the ions that make the region more acidic, right? So the high prevalence of these hydrogen requires it to having a low pH, right? So in the matrix, you have a high prevalence of this hydrogen, and these different shunting systems, these different complexes, create a very acidic intermembrane space in the mitochondria. This gradient causes the activation of this structure here called the ATP synthase for the production of ATP. So it wants to rebalance this osmotic gradient that is created, and one of the byproduct of this rebalance, in order to bring the hydrogen back into the matrix, it is the conversion of ADP to ATP. And so this is the main energy source that we're trying to always been looking for, right? And this is all created through the chemiosmotic theory, all right? And so when we look at the actual complex, there are 4 specific complexes that are important. And you remember from the TCA cycle and also the glycolysis, we have these reduced nicotinamide molecules that now need to be recycled back, right? And so the way they do it is they donate their protons and they become oxidized in each of these specific complexes, right? So the first complex, complex 1, you have a reduction of NADH to NAD positive, and you can see here that the hydrogen is transported down and into the intermembrane space, creating that chemiosmotic gradient, right? And so now, because this is created, there are these specific electrons now are being transferred in order to lead to the activation of these different byproducts, which is water. All right. So hydrogen is donated here, goes in. Then you go to the next pathway here. So you have complex 2 involving coenzyme Q. Right. The complex 2 has another dehydrogenase. Right. So both of these complex 1 and 2 have dehydrogenases, where the FADH2 comes in, releases its hydrogen, and allows for it to be transferred into complex 3 for it to be released into the intermembrane space. And again, you see the flow of this electrons. And these electrons are going, and eventually they reach this terminal region here, which is the cytochrome C. So cytochrome C is a compound containing a ferrous group. The ferrous group becomes oxidized, right? And these electrons are now transferred back into the creation of water, right? So hydrogen is not a very favorable ion to be present in either the intermembrane space or the matrix, right? So it needs to be reconverted to something that is less lethal, right? And so when this complex 4 now is releasing all this hydrogen in, right, you actually have a conversion of this transfer of these hydrogens into water, right? So you have a conversion of these molecules into water, right? And because of this gradient that is created still, there's too many hydrogens in the intermembrane space. A lot of it needs to be re-entered the mitochondrial matrix. And so the way it does it through the ATP synthase. So ATP synthase brings in the hydrogens, but because of this change in gradient, it leads to the activity of ATP synthase, which makes the ADP combine with the phosphate to give you a final product of ATP. Okay? And so this is just a very small understanding of the electron transport chains. Several drugs that are actually inhibitors of the different complexes, rhodonone inhibits complex 1, so you do not have this dehydrogenation of the NADH. And malonate inhibits complex 2, so there is no dehydrogenation of the FADH2. You have antimycin, so it prevents this efflux of hydrogen to go into the intermembrane space. Cyanide, so another chemical that you'll see a lot, prevents this formation of water, formation and the efflux of hydrogen into the intermembrane space. And then oligomycin, another chemical that's used as an inhibitor for ATP synthase, right? So these are different drugs, whether it's secreted by certain pathogens or certain toxins that we ingest, or even used as certain therapeutics for, cancers, right? And so all of these drugs have been used, and all of these drugs are inhibitors of specific pathways of the electron transport chain. So all of this is involved with the electron transport. Yeah, let's cross that out. Electron transport chain. So all of this is happening in the mitochondria, specifically the intermembrane, the membrane that is surrounded by the intermembrane space and the matrix.